Okay, our next lab technique is flow cytometry. This is basically a way to identify and count which cells you have in a population, usually in a fluid sample like blood, although you can also do it in non-fluid samples like a fine needle aspirate if you resuspend the cells in a solution first. So what kind of cells are in blood? Well, some of the most important ones that we care about are leukocytes like neutrophils, T cells, B cells, other granulocytes, or even hematopoietic stem cells. And can you think of any situations in which you might want to quantify white blood cells? That would be if you're trying to figure out if you have either too many white blood cells, which is called leukocytosis, or too few white blood cells, which is called leukopenia. So some examples of too few of a certain type of white blood cell would be HIV, in which of course you have too few CD4 T cells, or an immunodeficiency like Brutten's A-gamma globulinemia, also called X-linked A-gamma globulinemia, in which you have a deficiency of B cells. So when would you have too many of a particular type of cell? This would be something like a leukemia or a lymphoma. So flow cytometry is what you can use to count specific types of cells to either diagnose or monitor these types of diseases. Okay, so how does flow cytometry actually work? Well, we start out with our cells in a solution, which in most cases will just be a blood sample. Here you can see we have some red blood cells and some leukocytes. Then we incubate them with an antibody to whatever protein on the cell that we're interested in. So after we let them incubate for a while, we wash away the excess antibody. There's a good review of the proteins that can be used to identify different cell types in the immunology chapter. But some examples you should be familiar with are that all T cells have CD3 and either CD4 or 8, and B cells have CD19, 20, and 21. In flow cytometry, sometimes you might just look at one surface protein, so just one labeled antibody is all you need. But modern flow cytometers, which are the machines that you use for this, can often simultaneously process up to 9 different labeled antibodies at the same time. So you can be really specific with what you're looking for. For example, you can count only the cells that are positive for both CD3 and CD4, but negative for CD8. Okay, so once your cells have been labeled, usually you'll do another step to lyse all the red blood cells, and then time to run your sample through the flow cytometer. The flow cytometer is a machine that takes your sample of cells suspended in a fluid and guides them single file through a channel so it can look at them each individually. But wait, you're asking, there's like hundreds of thousands of cells in your sample. How long does it take to look at them all one at a time? Well, actually you can process thousands of cells per second, so it doesn't take that long. And how does the machine look at them? Well, for each cell, it shines a light on it with a laser, and then detects what wavelength of light is emitted. The trick is that the antibodies that you used earlier to target your cells of interest have fluorescent labels on them, which are activated only by specific wavelengths of light, and when they're activated, they emit a different specific wavelength of light. If you have 6 different antibodies, each one needs to be labeled with a fluorescent marker that responds to a different wavelength of light, so you can keep them all straight. So that's basically it. The flow cytometer then gives you data, which is what antibodies each cell had on it. Even cells that didn't have any antibodies on them will still be reported. They just count as cells that are negative for the proteins you're interested in. So flow cytometry is pretty cool, right? Okay, and more importantly, how will you be tested on it? Well, there's 2 ways. First of all, they could describe this whole technique to you, and then ask you what it's called. These can be a little tricky, since a lot of the lab techniques that you have to know for the step 1 involve labeling things with antibodies. So if they start describing something like, you suspect that your patient has a disease, so you take a sample of their blood and incubate it with antibodies that are conjugated to a label, then so far this could be flow cytometry, but it could also be an ELISA, which we'll talk about later. Only when they describe the process of running cells single file through a machine that detects them with lasers do you know it's flow cytometry. Okay, what's the other way you're going to see this in the exam? Well, they'll give you a clinical vignette with some clues about a disease, and then they'll show you a graph of flow cytometry data, and you have to know how to interpret this type of graph to get these points in your exam. So that brings us to our last part of what you have to know about flow cytometry, how the data is plotted. As I said before, you can have a lot of different antibodies labeling different proteins on the cell in one experiment, but you can only look at 2 at a time since the computer screen is 2-dimensional. As a bonus, you can also look at cell size, which is automatically measured by the machine without any antibodies required. So here's an example. Your patient has fever, weight loss, night sweats, and a rapidly growing mass. You get these 2 plots of flow cytometry data. Can you make the diagnosis? Take a minute to pause the video if you want to think about it. Ready? This is a tricky one without multiple choice, but the answer would be diffuse large B-cell lymphoma. The y-axis for both graphs shows cell size, and the x-axis on the left is CD19, which labels B cells, and on the right is CD3, which labels T cells. Everything to the right of the dotted line means that these cells have a bright enough fluorescence that we can count them as what we're labeling for. So these would be T cells here, and these are B cells over here. Everything to the left is something else. Notice how for the T cells you have only these relatively small cells, and that's normal because lymphocytes are small cells. But for the B cells we have two different populations, some large ones here and some small ones here. And there's a lot more large ones than small ones, which you can tell because there's more dots here. So in what condition would you have a bunch of large B cells? Well, that would be a large B-cell lymphoma. To get these right, I recommend going over the list of high-yield cell surface markers that correlate to different cell types, which are in the immunology chapter.